Closing this morning's session, we have Inge Holtman. She is an assistant professor at the University Medical Center Groningen, and she combines data science approaches to study psychiatric and neurodegenerative diseases. So please, the floor is yours.
Okay, good afternoon, everybody. First of all, I would like to thank the organizers for inviting me to speak here today. It's a pleasure. I'm the last speaker before lunch, so I hope I can still get your attention for the next 15, 20 minutes or so. Yeah, before I go into more data slides, I always like to start off a bit conceptual. And the fact of the matter is that if you look at a random group such as ourselves, we know that many of us will develop a brain disorder throughout our lifespan. Some of us will develop dementia, some people will develop multiple sclerosis, and other people might develop psychiatric disorders. And for all of those disorders, it is a big issue that if somebody gets diagnosed with such a diagnosis, we can really right now not predict how such a disorder will develop. For some people, it goes very fast. They have a lot of different symptoms, and for some people, it is relatively mild, and this heterogeneity is really largely unexplained. And I think there's a number of reasons for that. First of all, there's a limited understanding of genetic predispositions. GWAS studies have identified many variants in the genome associated with different disorders, but how exactly those genetic variants are influencing one's susceptibility and/or disease progression is still poorly understood. There's many cellular and molecular drivers to each of these disorders, as also discussed earlier today. But how exactly that influences specific individuals in their own unique disease manifestation is still not fully understood. And lastly, I really want to stress this point as well, is that the relationship between the clinical manifestation and the drivers, the biological drivers, is still not one-to-one. We know that many patients who are, for example, clinically diagnosed with Alzheimer's disease, if you do a post-mortem examination, you find out that it's actually not Alzheimer's disease. Moreover, many patients with brain disorders are suffering from multiple pathologies at once. So it's not as simplistic, you have Alzheimer's, so you'll have only this particular pathology. There's an interrelationship between these pathologies that manifest in a patient disorder-specific manner. And it's also one of the overarching goals of my lab is to study this heterogeneity. We try to unravel it by looking at shared and unique disease mechanisms across brain disorders, hopefully enabling more accurate diagnosis and prognosis. And in order to do this, I started a collaboration with the Netherlands Brain Bank back in 2020 for a project that's referred to as the Netherlands Neurogenomics Database. And what that means is the Netherlands Brain Bank is a unique institute. It was founded back in 1985 by Dick Swaap and has been able to perform more than 5,500 human brain autopsies since that time. And they have a massive amount of data. They have a lot of clinical information, they have pathological information. And most of these studies were relatively small scale, looking at maybe 20, 30 patients with one disorder, 20, 30 controls. And what the aim of this project is to start mining their collection in its totality. And we're using all kinds of AI, LLMs, image modality models to go from this wealth of data into clinically useful and to make it into clinically useful data resource that encompasses the multimodal data. One of the things I can talk about a bit for today is the clinical data, for example. This was published 2 years ago in Nature Medicine, where we used large language models to go from the original medical files of the NBB towards so-called clinical disease trajectories, encompassing 82 different neuropsychiatric signs and symptoms. And these symptoms are very valuable. They encompass cognitive domain, psychiatric domain, motor domain. And we've been playing a lot of data analysis tricks with them as well. We can look at these patients with Alzheimer's that were correctly diagnosed and compare them towards those that are misdiagnosed. What is different? We try to build AI models to do the prediction themselves. Do we still need a clinician? Well, it turns out we still need a clinician, but we were trying to see to what degree can we compete. Our competition wasn't that good, but it was a nice attempt. And we used the data to just look in a data-driven perspective into this heterogeneity across brain disorders. And I can talk a lot about this paper, but for sake of today, I would want to focus more on novel data. But it's good to mention these 82 psychiatric symptoms are very broad range. They go from fatigue, a symptom that's very important, multiple sclerosis. They go to all kinds of psychiatric symptoms, agitation, paranoia, psychosis, delirium, confusion, and each of them we clearly defined exactly how to score them. And based on these LLMs, we got a beautiful data set. And this is just looking at the psychiatric symptoms per se. And on the y-axis, we see the different symptoms: restlessness, mania, depressed mood. And on the x-axis, we see the different diagnostic categories, like patients with Alzheimer's, control individuals, multiple sclerosis individuals. And each dot corresponds to the proportion of cases in which we observed one of those symptoms. And what I hope you can appreciate is that the symptoms such as mania is almost identified in almost all patients with bipolar disorder and almost never in any other disorder. But then, for example, depressed mood is very common in bipolar disorder, in major depressive disorder, but also a considerable number of patients with any other disorder, going from Alzheimer's disease, multiple sclerosis, frontotemporal dementia, are also manifesting this symptom. And we really, at this point in time, don't understand why are some patients manifesting these kinds of symptoms and other people not. And can we get a better handle on this? Can we understand this from a genetic perspective or from a multi-omics perspective? And those kinds of studies we can now start to address with this wealth of data that were really impossible to deal with before. And well, there's even many more symptoms, and I can talk about my favorite symptoms for a long point in time. But I hope you understand that this is really a wealth of clinical data that is now added to the Netherlands Brain Bank. And this data gave really novel insights into this heterogeneity of brain disorders. And we want to continue upon this by also looking at the genetics information. And for this, we performed SNP chips. This is a workflow of the project. So again, we use the NND. On the one hand, we have genetics data, so SNP chip information, imputed to the genome. So we have several million variants across the genome. We've also been using large language models on the neuropathological data. There's extensive neuropathological reports. So we're implementing LLMs to also convert these neuropathological reports into standardized traits that we can use for downstream analysis. And then we integrate them, and we do a lot of different kind of analysis. We focus a lot on polygenic risk scores. So polygenic risk score is your overall susceptibility for any different GWAS trait. We look at the PRS symptom analysis, and we're also relating these polygenic risk scores and also GWASs towards specific neuropathological traits. So it's kind of a cool data set, right? First of all, the Netherlands Brain Bank, the name already says it, it's the Netherlands. So if we just look at like where do the donors actually come from, we noticed very clearly that most of the donors come from North Holland, even if you correct for the population size. So this is, this is a very clear overrepresentation, but we still cover the whole of the country, right? So you see on the population stratification level a really nice gradient going all the way from north to south. So people on the north are more common on the PC3, for example, and the southern individuals a little bit more common higher in PC1. And this beautiful resource allows us to take external GWASs, such as an Alzheimer GWAS, a Parkinson GWAS, a pure frontal-temporal dementia GWAS, etc., and start looking at like what do their polygenic scores look like in our unique pathologically defined cohort. And for example, if we do that for Alzheimer's disease, the quality of the figure really went down there. I apologize for that. I guess you can still read it carefully enough though. The point is that if you look at your Alzheimer's disease polygenic score and we compare control cases and our Alzheimer's disease cases, there's very clearly a massive overrepresentation of Alzheimer individuals for an Alzheimer polygenic risk score. Okay, well that's what you would expect, right? No novelty there. But what's kind of cool is that this polygenic risk score is also significantly higher in cases with frontal temporal dementia, another dementia that also shows a very modest genetic correlation with Alzheimer's, but also, for example, in cases with multiple sclerosis. And MS itself, the GWAS don't tend to correlate, but there's increasing lines of evidence suggesting that pathophysiological mechanisms are shared between Alzheimer's and multiple sclerosis. For example, one of our neuropathologists, Nina Franse, is investigating this in much more detail, but we know that tau and, for example, microgliosis, what Bart was also talking about earlier, are shared features of both disorders. So it could be that your genetic susceptibility to Alzheimer's disease, which is overrepresented for microgliosis and tau signaling, might actually also influence your likelihood of developing multiple sclerosis, which is an interesting hypothesis. And we can look at this on a global scale, and again, the quality of the figure is a little bit less than I was hoping. Maybe I should pay better attention at the PDFs. But what we can look at here is a whole number of diagnosis categories at the y-axis, and a whole bunch of polygenic risk scores. I don't have a pointer here. I think this one doesn't. Oh yeah, there is actually. Okay, so here we have all of the different polygenic risk scores. So this is the Alzheimer polygenic risk score in our Alzheimer's disease group, right? So it's very clearly significantly enriched. We see an enrichment of dementia with Lewy body. We see an enrichment of frontal temporal dementia. And here is the enrichment in the multiple sclerosis cases. And there's many of these very interesting associations. For example, we see that cases with frontal temporal dementia are also associated with Alzheimer's and Parkinson's and Alzheimer's. We see cases with motor neuron disease, mostly associated with ALS. We see that the MS cases are Alzheimer and MS. And we also, for example, notice that the major depressive disorder cohort of the Netherlands Neurogenomics actually shows a significant enrichment for bipolar disorder and not for major depressive disorder per se, which might be in line with the fact that we also observed that they have a lot of personality disorders, suggesting that they're a little bit of atypical major depressive disorder cases. And that's why it's so nice to look at these kind of aspects from different data modalities. We did the same kind of analysis for frontal temporal dementia subtypes like FTD-A, B, and C, motor neuron disease, FTD-tau, and there's many. The FTD field is very large. There's all kinds of frontal temporal dementia subsets. But here again, we see this sort of higher order profile that many polygenic risk scores are sort of acting combinatorially, right? So we see, for example, I think all of my slides went for very poor quality. I don't know exactly what happened there. But for example, this frontal temporal dementia A and B are very clearly associated with this Alzheimer polygenic structure, while C and this combined are much more like an Alzheimer or the FTD PRS. Well, for example, Pick's disease is very strongly enriched for frontal temporal dementia, but not for Alzheimer's disease. So that also suggests that these FTD subtypes are also maybe partially caused by differences in their polygenic structure. That if you have more of an Alzheimer type, it might influence to go a particular direction. And if your Alzheimer polygenic load is relatively low, you might go in a different kind of subtype. And what's also kind of cool, again, very poor quality, is that we looked at mutation information in this regard as well. And then even though the mutations with frontal temporal dementia are considered to be by themselves driving the disease, we actually show that the individuals who have frontal temporal dementia in the C9orf mutation have a significantly higher polygenic risk score. And then, for example, control cases or absent cases, unknown cases, or any of these other mutations. So that very clearly indicates that there seems to be an interaction going on between this familial mutation and your polygenic structure. And we can look at other kinds of polygenic structures as well. So these individuals are not only enriched for an Alzheimer's disease or for a frontal temporal dementia score, but they also have a significantly higher Alzheimer's risk score. They have a higher ALS risk score, but not, for example, a Parkinson's disease score. So at multiple levels, individuals who have a C9orf mutation have a very pronounced polygenic structure that most likely contributes to these people's diagnosis. And to tie my story back to the original starting point, right? We got all this symptom information. Can we now link the symptom heterogeneity of patients back to the polygenic structure of the individuals? Again, a poor quality figure. I stop apologizing. But what the figure shows here is that we're looking at Alzheimer's disease individuals, and we see the manifestation of different symptoms, right? So, for example, we see admission to psychiatric hospital, delirium, hallucinations, paranoia. So these are symptoms that are quite common in Alzheimer's disease, but also a large fraction of Alzheimer's disease don't manifest these symptoms either. And what we show here is that your personality traits, such as neuroticism and sociability, have a strong influence on whether or not you manifest these kinds of symptoms if you have Alzheimer's disease. So that's very clear, an association, and these two are also genetically negatively correlated by each other. And you can ask the question, like, is that specific for Alzheimer's disease, or does it also hold true, for example, in Parkinson's disease or in psychiatric disorders as well? And that does not turn out to be the case. In Parkinson's disease, there's actually an association with insomnia. And if you then look into the psychiatric cohort of the Netherlands Brain Bank, we see that hallucinations and delirium are actually associated with schizophrenia. And if I let that sink in for a while, I've been really digesting this figure for quite a while, I think it sort of suggests that the kind of symptoms that an individual manifests can be associated with multiple polygenic structures depending on the different kind of disease that you have. So if you have Alzheimer's disease, it might be more your neuroticism, sociability factors. And if you're in the psychiatric realm, maybe really this schizophrenia polygenic structure is the thing that sort of pushes you towards to manifesting this symptom. So different genetic structures can contribute to your likelihood of manifesting a particular symptom, which I find really cool. And also begs the question, like, how is this regulated molecularly? Can we use multi-omic signatures in the ICNS consortium to start tying these things together and identify what are the multi-omics, what are the cellular and the molecular mechanisms through which these kinds of symptoms are being manifested in the patients? So I think that's what the ICNS consortium, what we're going to work on. So again, it's really a pleasure to be here. This is still early stages, but I think there's much more really cool work that's going to be done within the realms of this consortium. And I hope you guys invite me again to speak in a few years. So that's pretty much it for my presentation today. But to summarize it, we established the Netherlands Neurogenomics Database in collaboration with the Netherlands Brain Bank, which I think is a very extensive resource to study brain disorders across clinical, genetic, and pathological domains. We've been using large language models to get clinical features out, to get pathological features out, and we show an association with the polygenic structure. They are both associated with different symptoms as well as with different disorders. And ultimately, I hope that we can tie it all together by integrating this into a multi-omics framework. And that's pretty much what I wanted to share with you guys today. Again, it's a pleasure to be here and looking forward to all kinds of discussions to have with all of you all in the break. Thank you.